All right. Hello, everyone. My name is Emma Holland, and this is my final video for Neuroscience 301. So the learned concepts that I thought were very interesting and I wanted to go over was neural development and deep brain stimulation. So learning about how our brains and the nervous system grows from babies to now was extremely fascinating. I really loved learning about the formation of our nervous system. Specifically, I was looking at the complexity of the neural tube and neural crest. The neural tube is when neural groove folds together and forms a complete enclosed hollow neural tube inside. The neural crest is as neural folds fuse. A population of cells separates from the tips of the neural folds, become a mass of cells. However, complications can arise within the neural tube. Neurulation is the process in the developing embryo that forms the neural tube, which leads to the brain and spinal cord. During early stages of development, if the neural tube fails to close, it can result in defects. So there are two different ways that these defects affect the system. The first is anencephaly, which is failure of the neural tube to spontaneously close at the cranial end, which means that the brain does not develop. The fetus basically develops without parts of the brain or skull, and almost all babies with anencephaly die within hours after birth. The second is spina bifida, which is failure of the neural tube to spontaneously close at the caudal end. The vertebrae overlaying the defect does not fully develop, which causes the vertebral arch to remain open, which basically is the spinal cord may protrude from the back. And this can be minor or it can result in paralysis. And surgery can correct some forms, but it also can be done prenatally. And I also thought it was really interesting that our brains continue to develop into adulthood. You usually think that children are developing within the womb and childhood, but we continue to develop way into our adulthood. Dr. Laura Boyd explained that neuroplasticity is when the brain continues to make new connections and prune away or replace old ones, which is how we continue to grow. And there are three different ways that it does this. The first one is chemical, which is transferring chemical signals between neurons. The second one is altering its structure through integrated networks of brain regions that function together to support learning. And the third one is altering its function because the brain shifts how and when it is activated. And there are actually whole networks of brain activity that are just shifting and changing. And according to Dr. Boyd, the most effective way to learn is personalized medicine and personalized learning for each individualized person. And I thought that learning about how our brain continues to grow was very important as it's very applicable to our everyday life. And then the second topic was deep brain stimulation. I was very fascinated by the research conducted by Dr. Terence Sanger and his student Jessica Vidmark. Ms. Vidmark explained DBS as a clinical treatment that sends electrical pulses into deep brain regions through implants and electrodes, and it seems to block the unhealthy brain activity from propagating through the brain. So basically, this electrical activity stimulates areas of the brain and it actually changes the way that it's firing. Her research specifically focused on children with movement disorders, and what makes her research unique was that she uses raw human data for an extended period of time. So she's collecting this data from many targets for stimulation and recording. And she talked about how she has a unique lab with a combination of many fields of medicine, neuroscience, and engineering. And her ultimate goal was to understand the connectivity of the brain and DBS itself, and to give patients the best treatment possible. Dr. Sanger specifically worked with patients with dystonia. And dystonia is when they have distorted movements and muscles that can be predicted, but not necessarily controlled. But by inserting electrodes, electrical signals can help to disconnect the malfunctioning signals in the brain. And just seeing and hearing about how this process has helped so many children was incredibly awe-inspiring and touching, and I was very grateful for the opportunity to hear their passion for their research. Some potential concepts that I wish we covered more in class was how neuroscience affects mental illness and creativity. So, being a psychology student, I'm very interested in studying mental health and with that mental illness and how we can help more people. Treatment and full recovery from mental illnesses has yet to be uncovered, and I'm determined to conduct studies and continue on how to make that possible. Neuroscience can help make treatments more effective and longer lasting. So, some early markers. Mental health symptoms are often present years before they're diagnosed through neuroscience called biomarkers. So, these biomarkers are biological indicators of medical condition or disease. In this case, the risk of a person developing a certain mental illness. And some examples of these biomarkers are the measured reduced volume of gray matter using an MRI and pattern brain activity using an fMRI. These biomarkers can also be used to show the common signs of someone with brain resilience, which can allow us to learn more about how the brain protects itself and grows within different people. Multivariate pattern analysis is a common statistical approach used to measure these biomarkers. And the article explained that we are able to promote mental health treatment through the identification of neural targets for intervention purposes. And we can look at what is being impaired within the brain for certain mental illnesses. Cognition, daily functioning, and motivation are all behavioral domains that are frequently impacted by mental health issues. By looking at these, we can try to determine the mental illness by which is being defective. Specifically, schizophrenia and bipolar have very identifiable affected regions in the brain. Brains with schizophrenia often have lower gray matter volume in frontal and limbic regions. The striatum is an important limbic lobe region affecting motivational outcomes for people with bipolar disorder. So, hyperactivation in the striatum is linked to an oversensitivity to reward and high motivation for rewarding experiences, so that dopamine. We can compare these neural targets or biomarkers with healthy brains to see the difference in these areas that are specifically being affected. So, our world has many talented and unique minds, and I was very interested in learning more about creativity and how that affects each of our individual brains differently. Anna Abraham studied the neurocognition of creativity and human imagination at the University of Georgia and has conducted many students on the subject. And I found a very interesting interview with her answering questions about creativity and her research. So, what exactly is creativity? She explained creativity has two important elements. The first one shows that our ability to generate ideas are original, unusual, or novel in some way. And that the second is that these ideas need to be satisfying, appropriate, and suited to the context in question. And then it's very difficult to define creativity because it's so subjective across so many fields. How can anyone judge it? And Abraham claims that we don't have yet enough tools to measure creativity. But in her book, The Neuroscience of Creativity, she discusses the 4 Ps, which are approaches to creativity. So this is a direct quote in which she says, approaches focusing on factors that thwart creativity may be external in that they are part of the environment, place, or internal in the form of traits and skills that typify the individual person. These are distinct from approaching creativity in relation to the mental operations that transpire during creative ideation process and the outputs thereof, the product. And there's more research to be done in the fields of creativity and neuroscience. But isn't that what's so exciting? The field of neuroscience is constantly expanding and growing with new research in all different types of subjects. And I absolutely loved learning about it all. I had an amazing class with all of you, and I want to thank Dr. Wilson for constantly teaching such interesting topics, and I'm just excited to learn more. Anyways, these are my references, and I hope you all have a wonderful winter break. Bye.